Machine learning is defined as the use and development of computer systems that are able to learn and adapt without following explicit instructions by using algorithms and statistical models to analyze and draw inferences from patterns and data. This is high-tech stuff, and it's dramatically changing the way that emerging biopharma companies are identifying and prioritizing potential therapeutic candidates and matching them with clinical indications. Further down the clinical continuum, automation, or the use of largely automatic equipment in a production process, is revolutionizing the way those therapeutics are produced at scale. On today's episode of the Business of Biotech, I'm digging into those topics with two leaders at Organicell, a clinical stage regenerative medicine company that's doing some exciting work in the therapeutic potential of extracellular vesicles, and more specifically, exosomes and microvesicles. Now, if you happen to be watching this pod on video, I don't want the youthful appearance of my guests to fool you. Both are pioneers in the burgeoning field of cell therapy. Dr. Mari Mitrani is a co-founder and chief science officer at Organicell. She also co-founded the American Cellular and Anti-Aging Center in Quito, Ecuador, one of the first autologous stem cell centers in South America. And she was instrumental in opening additional stem cell clinics in Guatemala, Trinidad and Tobago, and Jamaica. Dr. Michael Bellio is VP of Manufacturing and Processing at Organicell, responsible for research, development, and manufacturing of novel biological medicine for the treatment of pulmonary, cardiovascular, and orthopedic degenerative diseases. For his part, Dr. Bellio has developed a methodology for the large-scale manufacturing of exosome products from bioreactors for use in translational and preclinical research. With that intro, Doctors Mitrani and Bellio, welcome to the show.
Thank you so much for having us.
And I'm honored that you could join us. I want to start with you, Dr. Mitrani. This isn't the first time that you and I have talked, and we last spoke, I don't know, maybe a month or so ago. You told me a story about launching Organicell back in, I think it was the 07-08 time period, back when cell therapies were burgeoning, yet they faced roadblocks due to political and public perceptions around stem cell sourcing in particular. And I want to...
Specifically in the U.S., yeah.
Yes, in the U.S., right. And I want to remind listeners that they can read about that story at bioprocessonline.com. But I want to kind of go back to that time frame, Dr. Mitrani. And back then, the focus was kind of on research, and it was largely happening in academia. But I'm curious if fledgling companies like yours and others in the space were, at that point, leveraging machine learning or artificial intelligence or compute power of any type to any degree in those early days, and if so, how?
Yeah, so actually, because we were really into autologous stem cells, we did transition from your regular puncture to extract those stem cells from the bone marrow to a device, an apheresis device. And that was really the leap that we had from an just manual injection to be able to collect those stem cells from the bone marrow. And we were super futuristic at that time because we were not making punctures through the iliac crest. We were doing sternum punctures, which made the whole procedure less invasive. We didn't need DOR. It was a local injection with anesthetic to be able to collect those. And even then, with that protocol, we were able to jump into a more technical and more precise collection of stem cells through an apheresis device, where we were just input in the computer what specifically we needed, and we will collect from the free-flowing blood through a process that lasted between 3.5 to 5 hours, where the device will collect 30% of the free-flowing blood of the patient outside in the device, and through the computer and through a Daltonic weight, it will just collect the stem cells that were in the free-flowing blood. So at the end of this 5 hours, 3.5 hours, we will have a bag of just specific stem cells that were floating in the free-flowing blood, and therefore we didn't have to manually extract them, right, and then centrifuge them. this device will do everything for us. So even in the early stage, we were pioneering in new techniques where we will incorporate more like not artificial intelligence, but device or computerized device that could pinpoint specifically those stem cells that we wanted.
Mm-hmm. Yeah, and I'm curious about when. Well, let me start with you, Dr. Bellio. Let me just run over to you for a minute. When did you join Organicell?
So I started working for Organicell about 2 years ago.
Okay.
I trained. I got my PhD at the University of Miami in the molecular and cellular pharmacology program. From there, from grad school, I did my postdoc and was an assistant scientist in the Interdisciplinary Stem Cell Institute, where I learned to do a lot of the cell manufacturing within their GMP for all their clinical trials that were ongoing. So while I was completing my projects there, I was working on these large-scale expansion techniques for the mesenchymal stem cells and other types of primary cell lines and how to make exosomes from that large-scale batches. Mm-hmm. So that was my kind of first introduction into the exosome world. And that was where I kind of started to link up with other people that were doing extracellular vesicle research in the Miami area. And I met Dr. Mitrani, and it was the perfect match. It was, Organocell was, had this vision and this goal of developing these new therapeutics with exosomes, building a facility where we can complete all of our clinical trials, basic science research, everything in one place. And that was really right up my alley, doing that translational research as well as manufacturing products for clinical use.
Yeah.
So that's what kind of forged our initial relationship, and it's only been, excellent from there on.
So you bring an interesting perspective because it wasn't too long ago that you were coming out of academia and into the,
Exactly. Yeah. Right. It was the best experience for me. And working at an academic CGMP facility was really the key for me because, research is still really a big part of developing these new clinical therapeutics. But, you're so busy in these GMPs, you can only do so much research. So it's all about targeting the research to get those precise questions you need to really prove that your novel therapy is worth the clinical trials, prove to the FDA that you got the safety, efficacy, and the mechanism of action. Mm-hmm. And so it was really an important start for me to remember that academic research route as well as, towards the industrial manufacturing of a drug.
Yeah. Can you, it's sort of an aside, can you kind of compare and contrast for us the degree to which compute power, machine learning, whatever you want to call it, artificial intelligence, mm-hmm. right? Computing, the degree to which you relied on or used or learned compute power in biopharma and cell and gene, research, cell therapy research in academia versus, coming on board at Organicell and kind of working in industry.
That's an interesting comparison. I mean, they are very similar because in academia, when your lab or your group is working on clinical trials, it's essentially the same requirement that a biotech company would have. You need to demonstrate what are the molecular components of your product and use that high-powered bioinformatics analysis or that high-powered computing to help kind of predict why is it an effective therapy and why would I want to use it for a particular disease? I think the biggest difference with academia in my experience was you can be a little bit more exploratory in some of like the basic research labs that are doing a lot of computing and bioinformatics for research projects to really fine tune, some of the molecular links and pathways and all that. Where in biopharma at Organicell, we're really focused in on our clinical trial development. So all of the computing and the development is all kind of centered around that one goal of advancing our therapeutic purpose and really demonstrating why our drug is effective for our different therapies and using those bioinformatics softwares to really help build, a solid basis for why we're doing what we're doing and why it's effective.
Yeah, well, that's good. Let's kind of hover there for a minute and tell me Dr. Mitrani, maybe this is a good pivot back to you, because it predated Dr. Bellio. When did Organicell, like as it relates to using bioinformatics to further the clinical effort, when did Organicell kind of go all in and really buy into digital transformation or bioinformatics? What did that look like?
Well, I think it's always been with the field. It's not like we pivot and it was like a before and after. As I'm mentioning, since the beginning, we have always looked into what is the cutting edge and state of the art things that we can incorporate into our protocols. Remember, Organicell or our what we started, our companies are private companies that started and brought into the public company where we are right now, was always clinical focused. So even though we had the research as the most important thing, but clinical and finding solutions for the patients was always our goal. So now in the bigger scale of manufacturing, when we transition from autologous stem cells to perinatal derived stem cells, and there furthermore, when we transition from stem cells to exosome derived therapeutics, I think that brought in a lot more of this AI or computer related development, because, first of all, exosomes were recently just discovered, let's say in the scheme of this last 50 years, right? Even 10, 15 years ago, we did not know about this exosome new flourishing field. And now we're super embedded to this. It's been 4 or 5 years that we're really into understanding what is. So now that there's devices that can let us go into looking what is inside of these exosomes, right? Because before we just thought it was excretions of the cells, because we didn't have any technology that would allow us to go inside of those exosomes and see if there's actually anything there. Scientific and medically, we thought that was a secretion from the cell. Until recently, we were able to, there's devices that actually see how is it formed, how is the exosome formed, and most importantly, what's inside. So I think that was like our biggest breach into the AR or computing, to actually find from our final product, after everything that we do, finding and characterizing what's inside of those exosomes. And I'm going to let Dr. Bellio go through that one, because that has been our passion and what we've developed and what we put all of our heart and our brains into it. So.
Yeah. And, in fairness, obviously my understanding of exosomes is entirely layman, right? Especially compared to yours. But since I spoke with you last, Dr. Mitrani, I've done some reading, and from what I gather, we've learned a lot, but there's still so much to learn around the function of exosomes and their potential as a therapeutic vehicle. So, Dr. Bellio, I'm curious how. It's a sort of a big open-ended question, but how do you anticipate leveraging bioinformatics to help gain that understanding that not just the entire industry has yet to see?
Yeah, no, so bioinformatics and that technology of using this computing to help us kind of characterize our drug component, our exosomes, has been critical to the start of our clinical trials pipelines that we've developed within the past few years. And one of the great things about our therapeutic is Organicell has developed a novel source of the exosomes. So the traditional kind of research path of exosomes has been in the cell therapy field. A lot of clinical trials were testing mesenchymal stem cells and different primary cell lines, and they're getting kind of inconsistent results with larger scale clinical trials for one reason or another, because cells are very sensitive, the viability can be an issue, and they were noticing that very soon after you transplant cells into someone, they die off very quickly. So even though there were positive results from these cell therapy trials, it was kind of interesting to think of, well, if they die off very quickly, what is really the mechanism of the cell-derived therapeutics? And they realized that the mechanism is largely linked to the secretion of these exosomes. So that kind of led to this new avenue of exosome research, as in, well, if we can figure out ways to extract the exosomes rather than the cells, we can use that as a therapeutic instead, because exosomes are way more stable. They're, seem to be safer because it's not a live cell, and there's no real requirement for cryopreservation reagent, which can also kind of complicate the preparation of cell therapy. However, so the natural transition of studying exosomes kind of started with, exosomes from mesenchymal stem cells or from these primary cell lines that were already being used in cell therapy. So there's wealth of publications and scientific research about that type of exosome. However, Organicell was, experienced and were previously looking into perinatal tissue sources, because perinatal tissue is, a very good source for therapeutic medicine because it's generally considered trash tissue, and you can collect it from healthy consenting mothers with no risk at all. And all that, the features of these perinatal tissues, because they're from young tissue, they seem to have, more of a therapeutic potential. So that was the arena that Organicell had, kind of set itself into. So then, before my time was when Dr. Mitrani and her team were trying to pinpoint what features of the perinatal tissue was really the best therapeutic, component, and what would make the most sense for a large scale distribution, clinical trial development type of thing. And that's where they started. That's where they discovered this idea of using amniotic fluid and perinatal tissue derived extracellular vesicles. And I love that because it's a very novel source of exosomes. There's very little literature out there about what are the components of perinatal tissue secreted extracellular vesicles, what makes them different than the stem cell derived exosomes. Because an exosome is essentially just a hollow lipid shell. And what's inside the exosome, the microRNAs, the proteins, the cytokines, are really what determines what's the therapeutic value of that exosome. So it would, the first thought is, of course, these exosomes are very different than a mesenchymal stem cell or a stem cell cultured exosome. So the first and most important thing for Organicell to do was to figure out what's inside these perinatal tissue secreted exosomes and why would they be good for all our clinical applications, for COPD, for osteoarthritis, for all these other degenerative diseases in our pipeline. What is it about it that makes them better or different than a stem cell derived exosome? So we did that. Our research has really been focused on using bioinformatics and large scale sequencing, proteomics, mass spectrometry to really characterize every component of that perinatal secreted exosome. And we've used these softwares to help kind of build our networks and predict those microRNAs most likely to be involved in lung repair or anti-inflammatory conditions. And it's really been, critical and a very important part of our submission of these IND applications to the FDA to really kind of bring our, theories to the forefront and help guide our basic science research as well.
Yeah, it's exciting stuff. So another two-part question for you: what would you consider along the way to be Organicell's biggest, bioinformatics-driven win, if you will, in the development of the company and your clinical effort? And then two, how would you characterize the adoption of this kind of technology in the greater space? Is it common, or is it still sort of a competitive differentiator for some?
So the first part, our biggest win was being able to really sequence all of the microRNA and protein cargo within this perinatal secreted exosome. Because, like I said, the source of exosome is so different than the traditional cell culture expanded exosome. Mm-hmm. That was really the most important part for us, was to be able to characterize from high expression to low expression, what are the microRNAs in this? And we used all of that bioinformatics to help guide our the writing of a lot of our clinical applications to the FDA and demonstrate, what is in this product and why is it effective? And and it all kind of trickles down from there. Once you have these, real portfolios of lists of high protein concentrations, the microRNAs, it's really guided all of our translational research as well for our animal studies and for all of the publications we're working on, of course. It's how we're able to kind of predict these mechanisms. And that could be one of the hardest parts about this exosome therapies and cell therapies in general, is everyone wants to know what is that one mechanism that really triggers the regeneration or the healing. But in reality, there's lots of pathways that are affected by these extracellular vesicle products. There's lots of microRNAs that could interact with different types of cells. So it's really important to try to think about what are the cellular targets of our therapy and what could be the main hitters and the main links to what we're seeing in the clinic. Mm-hmm. And then the second question you asked, can you repeat it again?
Sure. Yeah. It kind of, so, it occurs to me that if you're first or early to the game, this is a competitive differentiator. So I'm just curious about your perception of sort of how this technology is being adopted, in the greater space.
Yeah, no, I think in general, you see more and more bioinformatics and sequencing and proteomics being done because all these exosome therapeutics are so new, and dependent on how you make your exosomes really dictates what's inside it. Mm-hmm. So, it's almost a requirement at this point for when you're developing these new therapies is you need to use bioinformatics to help guide your initial kind of thought process of, that mechanism of action that the FDA wants to know.
Yeah.
And really to kind of show, what your new therapy can do. And especially in the biopharma space, that's the bioinformatics is probably, it's really critical because all this gene editing and all these new vaccines that are now RNA based, you need these conformational things to kind of suggest, or what could be an off-target effect as well. You could predict all kinds of interactions and stuff like that you need to know.
Yeah.
But I really think that this is like the biggest win for Organicell is our early on development on this trying to find the pathway, right? We're used to a biologic or other kind of medicine, like one compound and then trying to track that one compound throughout the body. How does it absorb? How does it metabolize? How does it get secreted, right? When you're talking about biologics and a product like we have that is so vast of either exosomes and proteins, it is a little bit more difficult to just try to radioactively target one component and then track it throughout the way of the pharmaceutical processing more than everything that we have, right? So I think that we it's been our mission to really try to see how far we can go, how deep we can go into knowing what is in there and why would it work. And as Dr. Bellio was saying, it's, we've been working with FDA for over a year right now, and with 3 IND approvals and 18 EIND approvals. And FDA, when we have our calls with them, they're extremely open to work with us. We collaborate really well because our background and all of the scientific theoretical part is there. It's we've worked a big book of knowledge on why and how, right?
Yeah.
And I think that is a big difference that the competitors will have. And as Dr. Bellio was saying, I think that the whole field is moving this way. I mean, every year that we participate in the societies of extracellular vesicles and gene and cells therapies, the entire field is moving this way. So that's where we are all going, and using bioinformatics or AI into our either diagnose or production or mass production. It is the way that we're all moving.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech.
That's a great segue, Dr. Mitrani, to, where you guys are on the clinical path and your production scale. There's, as I mentioned in my intro, there's plenty of opportunity for automation, as companies ramp up clinical supply and even beyond into commercial production. So I'm curious on your opinions on when companies like yours should begin thinking about and planning for process automation, beyond bioinformatics in the discovery phase. When is it time to start planning about how to apply automation technology, process automation technology, on the clinical path?
Yeah, I mean, I think it's never too late to start thinking about automation, because at the end of the day, as your production grows and grows and grows, you're going to need some kind of automation, because people can't do everything. Like, it's a lot of work to do some of these processes. It can take all day, multiple employees, and there's a lot of note taken and records that need to be kept throughout the process to ensure, safety, sterility, that the steps are done properly. And automation really kind of helps reduce error, keeps consistency, and things like that. So there's all avenues in a cell therapy manufacturing facility that automation could be useful. The most obvious and probably the easiest way to start with automation, and is how we're starting with automation, is with product filling. Mm-hmm. Like, to make sure, when you make large lots of product, it can be extremely tedious to fill hundreds of vials of product by one person, to make sure the vial's filled, capped, labeled, and everything's done sterilely. So manual labor is a lot. So automation is one of the fastest, easiest things to get in there, to kind of set up a system that you can automate a 1 ml, 2 ml fill of your vial, whatever you want, and then crimp it, cap it for you, and then you're done. And that could save hours of manpower and really ensure the accuracy and the consistency of that stage. And I know from other companies and other things, automation or digitalization of records, like batch records. Every time you do a lot or a production, you need to have this thing we call a batch record, which is ultimately the collection of every piece of the production components, from donor qualification to manufacturing to the fill-in to storage and then product testing. Those batch records are the soul of that product, where the quality and everyone can review that. There is some level of automation and digitalization that you can incorporate in maintaining those records. Ways that, if you're able to automate the input of data throughout, manufacturing, you could program in a step that would alert you or alert a manager if there was a discrepancy, or if a step was skipped, or there was something wrong with the volume or something like that. Mm-hmm. Automation could really be useful to kind of pinpoint in real time errors in your manufacturing process that might not have been caught until it's too late.
Yeah. Yeah. I can see where it's tied to, as you said, data integrity as well as product quality. And I imagine I'll look to you both to give me some anecdotes around this. I mean, in any biologic production capacity, those are very important things: data integrity and product quality. The supply chain that Organicell endeavors to create and take to scale is especially unique in that, to your point, you're harvesting perinatal tissue, and taking that all the way through to a biologic therapeutic product. So what are some, unique to Organicell challenges around that dependence on perinatal tissue and maintaining data integrity and quality of product that you've faced, and how has technology helped you address those challenges?
Yeah, so you're right. So one of the biggest challenge of biologics is the source material. And donor qualification is probably one of the most important aspects of our production. Organicell, we have a medical director who qualifies the donors for each one of our production. And one of the biggest challenges is that donor qualification needs to happen relatively quickly. Mm-hmm. You know, because all these tissue donors, they're not really pre-planned to donate the tissue. It's really these mothers that have a healthy full-term pregnancy, you know, go in for their C-section surgery, and at that moment they consent to, you know, donate in the perinatal tissue. So a lot of these systems we set up, you know, ahead of time. We have a qualified, you know, site, a qualified hospital that can handle the tissue. They know how to collect it, and they have on site all of the donor qualification materials that a donor needs to fill out. And after they agree to consent to donate, they'll fill out these surveys, the questionnaires, so that we can determine if their social medical histories fit our criteria. And then they also get their blood drawn so that we can run the virology panels. And from that, you know, everything happens really quickly. The tissue gets collected, it gets shipped to us overnight, you know, on ice, fresh. And then their blood sample gets shipped to a virology or a CLIA certified testing lab. And the viral testing is really probably the first place where this digital requirement comes in, because we need to be ensured that these testing sites get the blood, they have the right test orders in place, because a lot of different companies have different test requirements that don't necessarily meet Organocell's requirements. Right. So we need to make sure that digitally that test facility has all of the different viral panels that we require ready to go, and then make sure that we get the test results back as soon as possible. Because ideally, we want to be able to process the tissue as fresh as possible when we receive it. And it usually takes about 2 days to get some of these viral panel tests back. So within that 2 days, we have to make the decision of, well, do we want to use this donor, you know, to make the product or not? Because if the donor doesn't qualify, you could potentially, you know, lose money and lose reagents and materials because you processed it and you ultimately can't use it. Mm-hmm. So we've invested, you know, in our flow of our workflow to really kind of use that digital interface with the testing company to give us the medical record testing, all of the virology panel as soon as possible, and then deliver that to our medical director so he can make those, you know, decisions about whether this donor can be used or not. You know, and that's really a very critical and challenging step, you know, to get all of that in line really fast. And it will continue to get even more challenging the more product you want to make, the more donors you need to be qualified. Right. Because, you know, that's another challenge with biologics is, you know, you can only get so much product from one donor. So every time you have a new donor, you need to keep that, you know, qualification stringency as strict as possible. And moving as fast and smooth as possible too.
Yeah.
But I think that we've been super proactive to move ahead with this trend of us our production moving in larger scale, right? So besides everything that Dr. Bellio said, we do have an IRB approved donor qualification. So we've always been one step forward and one step ahead of the curve, even though FDA and American Association of Tissue Bank have established what are the rules and regulations for a donor to become a donor. We also have added this one more step of making sure that there's an IRB approved donor qualification. So we ensure that those donors, and that is, I think, the most important thing on this field, because we are working with donors, that those mothers are healthy, those mothers of babies, that their social, economic, and medical history has been completely checked, besides all of the virology that Dr. Bellio mentioned, right? So for us to ensure that every single product and every single batch that we create is always consistent on the final product is always consistent. It can give us that clinical effect that we're looking for.
Mm-hmm. Yeah. Yeah, so let's. Man, I feel like we could talk about this particular thing for a long time.
Hours.
Yeah, but we do need to keep the conversation moving. So I want to shift a little bit and talk about your clinical program. I believe it's your lead clinical program in COVID-19 and COPD therapeutic, Zofin.
Yes, Zofin is our lead product. And the one that we were already trying to do so many clinical trials before the pandemic hit, and what led us pivot into this lung injury that we were learning, investigating, and getting results already that allowed us to present this information into and for COVID.
Yeah. So let's talk about how you are currently leveraging automation and plan to further as that clinical trial progresses in the production of Zofin. So you're manufacturing in-house. Do I remember that correctly, Dr. Mitrani?
Correct. Yep. That's 100%.
Okay. So as you scale up to meet further clinical needs, paint the picture for us around what points of automation you're most dependent on in that scale up.
Yeah, I would say right now our most dependent automation is the vial filter, the vial filling step, because with our clinical trials, I mean you need a lot of doses to do all these clinical trials, and you need a lot of doses to do our research as well. So that has kind of helped us reduce the manpower hours of our production time. Because we're using fresh tissue, all of our manufacturing is usually done within 1 day. So it can be a real long day without some of this help.
Yeah.
So the vial filling is really one of the first things that we've started to incorporate in our production scheme that has helped us manufacturing wise.
But from that point, SOPs and as he was saying, all of that donor qualifications and everything else goes hand to hand in the digitalization or automation of the production. So
yeah, because I would say, like, as I was touching on upon earlier, the batch records and the paper documents of the cell therapy or GMP production is really heavy. So being able to digitalize a lot of those records, the worksheets, the different stages of production has been kind of helped us, not only with organization, but it's really important, ways to kind of store important data on, for our group, and to really organize which processes to follow one after another. Because after we manufacture the product, there's a whole nother there's a lot of molecular analysis we need to do of each lot product. We do like characterization of every product that we make. We do a nanoparticle analysis of the exosome size, concentration. We do different protein analysis to show, to measure that the protein quality of the product is maintained. Hyaluronic acid is one of our important mechanisms that we're interested in our product. So we have to qualify the concentration of that each time. And being able to store a lot of that information digitally with these batch records and these worksheets, has kind of helped to better streamline the review of those processes and to collect that data and to analyze it sooner than later than looking at it manually. So yeah, that is kind of a different kind of automation that has really been kind of important to handle a lot of products at once.
Sure. Yeah, Dr. Mitrani, the way you sort of described it a minute ago is it's like a workflow, if you will, or like a digital kind of workflow. Did you? Curious. Did you architect and build out this workflow in-house using Organicell folks, or did you outsource? You turn to like some IT propeller heads and outsource some of this architecture?
I mean, when we started, it was more trying to go outsource it. And because we were trying to shoot and see what is it that we want to analyze. But soon after, we brought it all in-house, bought all the machines and the devices that we needed, because we knew specifically what we were looking for, and therefore what we could do in-house. And being able to have the team that is so qualified to be able to run this processing also as fast as the processing is done for every batch, we have our samples in mid-processing and at the end of the processing to know where we are sitting at and how we're doing, right? So all of this depends on many devices. It's not one, it's not two, and they're like the ones that are looking for actually the size, the nano side, right? The ones that can actually look for the biomarkers that are outside of the membrane, the ones that are counting, as he mentioned, the amount of proteins that we have at the final, or our hyaluronic acid, what's the concentration on that? And even endotoxin, knowing that we run a procedure throughout the whole day and knowing that our final products don't have any endotoxin without relying on, many vials will go out for private testing for that, but us knowing that we did the correct thing in-house, it is very important to, I think that was our biggest strength, making sure that we created a team and created a facility where we can automate most of the things that we are looking for.
Mm-hmm. Yeah. Are you anticipating at any point outsourcing to a CMO as you approach commercial, I guess, commercial scale, or is it too early to say?
Do you want to take on that?
We're ready to work with CMO collaborators if needed. All of that workflow that we developed we could easily integrate a CMO into that workflow.
And that's why I asked, to be clear. To be clear,
absolutely. We're at a point that we could, and we do everything right now. But even if we were working with a CMO collaborator, I think that integration of this digitalization and this automation would be even more important, because I would envision that Organicell, would still be highly involved, and we would have to still do donor qualification. And a CMO couldn't do all of the tests that we do on all of our lot products. Like Dr. Mitrani was just saying, we invested a lot of money and time into developing and picking those machines and those processors that kind of analyze the exact things we need for our clinical trials. Like everything that we need to analyze the components of the exosomes, we pretty much have it. Mm-hmm. So we don't need a CMO to do that. We don't need to work with the CMO to analyze and qualify our product for clinical use. It would only be, we would collaborate with these people that help the large scale manufacturing process. And throughout the way, Organicell would be able to monitor the production steps, determine if everything was being done correctly, and then to really determine whether that final product met our standards of clinical use and release it for the therapy.
Yeah. Okay. Well, good. We're, like I said, we're running short on time here. I want to thank you for your transparency and sharing your, the opening the kimono, if you will, on the adoption of bioinformatics at Organicell. It's been super enlightening. But before we wrap things up, I do want to be fair and give you guys an opportunity to share, any updates on the company, on the pipeline, on Zofin. I know you've got some preclinical work going on too. I don't know if you're ready to talk about that, but the floor is yours to share with us, what's going on with Zofin and beyond.
Yeah, well, it has been a phenomenal year for us. It's been a roller coaster, but a really happy roller coaster, I would say. Just the opportunity of being able to explore our product, our therapeutic, into other fields that we were not even expecting it, but doing it so with all of the scientific data behind it, and now understanding that even if COVID does not last forever, we do know how to transition into those other realities, right? For example, the long hauler is one of the populations that I am personally very passionate about, and I guess the whole team, all of our medical advisors and the team that is working with us for that, because it's most of the companies have focused on the preventative side, right? And thank God now we have a vaccine that it's rolling out, and that hopefully COVID will be going down now. But there's still that population of the long haulers that has not been addressed. So we are working really hard to see if that will be our next avenue to propose a serious therapeutic proposal, right, for our therapeutic. And then again, now finally coming up around that 1 year and being able to have the approval for our COPD trial and getting that COPD trial going. I will say in a couple of weeks, we will have some good news to share with everybody. We're a public company, so we can only share what we can only share. Sure. But nevertheless, COPD is one of those degenerative conditions that does not have a solution. And it fuels us, and it believe me, that is what makes us work so hard trying to find a solution or to improve the quality of life for those patients that have no solution in regular medicine, right? So that is COPD. We are working really hard with our osteoarthritis preclinical data, coming up that as well. So the pipeline is moving along strong and steady, I would say. More than just stay tuned. We're working hard. And as I always say, people forget about the people that work behind the scenes on the therapeutic, the preventative. And as the scientists, we're here, we're not stopping. Science will show up. We are not resting. We are here to try to find solutions and hope for all the people that are in need.
Yeah, well, those people in need appreciate the work you guys are doing, and I encourage you to keep up the great work. I mean, it's really exciting stuff that you're working on.
Yeah, and I can speak really quickly about what's next in line for our research and development at Organicell.
Yeah,
and I said one of the best things about, or one of my favorite things about the company and the lab we've been building is we really are very invested in the research and development and trying to figure out how to make with the therapy we have now even better. And we do a lot of animal studies with different lung models because, we've kind of started, a lot of these investigations about how the perinatal secreted exosomes can help suppress inflammation and help repair lung damage. And all that bioinformatics data we've collected is a wealth. It's really a treasure chest of things that has kind of led us into all of our different projects. So we're really working on figuring out, what are those main microRNAs that are involved in the regeneration that we're seeing? Because one of the natural evolutions of the therapy is also looking at the engineered exosome. How can we make these exosomes better? We know we're demonstrating that they're safe. We're demonstrating that they're potent. But how can we make sure that how can we personalize the therapeutic for different diseases? And engineering the exosomes is one avenue that we're looking at. And to use that bioinformatic data, really prove which components are the ones there and overexpressing that in exosomes. And that could be the next generation of therapies that kind of come in line.
Awesome. Well, we'll be paying attention. I guarantee that. You guys are an exciting company to watch. I mean, Dr. Mitrani will tell you, I interviewed her a month and a half ago, and then within weeks, I was like, you got to come on the podcast, So I'll be back, and hopefully there'll be some good updates coming out of Organicell, throughout 2021. So we'll be paying attention.
Awesome. Great. Well, thank you for having us.
Oh, thanks for joining me. I really appreciate it. So that's Dr. Mari Mitrani and Dr. Michael Bellio. I'm Matt Pillar, and this is the Business of Biotech. We're produced by Bioprocess Online, part of the Life Science Connect community, in partnership with Cytiva. For its part, Cytiva offers a deep well of content designed to support new and emerging biopharmas at cytivalifesciences.com/emerging biotech. That's CYTIVA. lifesciences.com/emerging biotech. Check that out. Go to bioprocessonline.com, subscribe to my newsletter, search up Organocell, and read that article I wrote about Dr. Mitrani and Organocell a little bit ago. And if you like what you're hearing on this podcast, subscribe, give us 5 stars. In the meantime, thanks for listening.